Alrighty, everyone. Good morning, good afternoon, good evening. This is Dr. Kaz, and this is the final part of Chapter 22, the immune system, our review of the immune system. So I'm going to pick up where I left off, and we were talking about our lymphocytes. And we just finished talking about our B lymphocytes and how they will differentiate into plasma cells and produce antibodies. So we're going to move into what we call lymphocyte recirculation. And when we're talking about lymphocyte recirculation, we are going to be discussing what happens to these cells, these B cells and T cells, all right, over time and how they move around. Now remember, excuse me, the majority of your immune cells are going to be found in tissues and not in circulation. And so some of you might be familiar with the saying, if you want to get out there, if to meet somebody, you got to get out there and meet somebody. Usually this is when folks maybe have been dating for a while and they had a long-term boyfriend, girlfriend, what have you, and they broke up and they want to have a significant other in their life. And so their friends are like, get out there, go meet somebody. So this is basically what happens. These lymphocytes are going to leave their comfort zones or these secondary lymphatic structures like a lymph node or the spleen. And so they'll just get out there and they'll enter back into circulation. And when I'm talking about circulation, I'm talking about both blood and lymphatic circulation. And so they'll just run through both of those circulatory systems there and wander around. And so what they're going to be doing is they're going to be looking for that specific antigen. So it's much easier to come across that specific antigen if they put themselves out there, if they put themselves into circulation. And so that's what they do. And so, of course, they'll move around. They might reside in some other secondary lymphatic structures, but that's what they're just going to wander around, maybe take, down in Australia, they call it a walkabout. Well, this is the lymphocytes version of a walkabout, when they go out into that recirculation in search of that specific antigen. So coming down here to the last part of what happens with the lymphocyte when it encounters an antigen, we're going to now talk about the effector response. Specifically, the effector response at the site of infection, right, where those pathogens, all right, have that's their base of operations. And so the effector response, we're going to talk about the different various mechanisms that these cells use to get rid of the pathogen or to eliminate the antigen there. So our helper T cells, those are the CD4 cells, we've seen before how they release interleukin-2. The interleukin-2 has various effects that we've already talked about, how it can self-stimulate the helper T cell, and then it can cause activation of other lymphocytes here. But in addition to the interleukin-2, we'll see other various chemicals, or what we call cytokines, that these cells are going to release. That's why these cells, they're so crucial, because they're not only going to regulate the cells of adaptive immunity, they're going to regulate, all right, the cells of innate immunity. And some of those cells, remember, are natural killer cells, for example, right? These cells will help to activate those natural killer cells to do what natural killer cells do, and that's to kill, right? To get rid of pathogens, right? To destroy whatever it is that antigen, that foreign antigen is. Then our CD8 cells, the cytotoxic T lymphocytes, right? Their job during the effector response is, of course, to go after our unhealthy cells, but they do so through the process of apoptosis, okay, that's cell death. And when we're talking about unhealthy cells, just to remind you, unhealthy cells include infected cells, either by a bacteria, virus, parasite, what have you, or just cells that might start to display abnormal receptors on their cell surface, because yes, they may have been infected, but maybe they're undergoing some neoplastic change, right? The dreaded C word, cancer, right? Or just cells that are just starting to get old and not working correctly. They'll come in and take care of them. And then finally, our plasma cells, which come from our B lymphocytes, and their job is simple, right? Plasma cells make antibodies, make those immunoglobulins, get those things out there to help us, all right, to get rid of whatever it is, all right, that those pathogens are. So let's get into a little bit more detail. about the effector response with our T lymphocytes, specifically the T helper cells. So we know the same old story, right? These cells, once they've been exposed to the foreign antigen here, right, they now have become activated. And so a couple things will happen. These cells will proliferate and they'll make more activated T helper cells. And sometimes, right, we'll get the production of our memory T cells or our memory helper T cells. Again, those memory helper T cells, in case we come in contact with this antigen at a future date, will help to initiate the effector response quicker. So these cells are going to do what they love to do, and that's to release chemicals. All right, these cytokines are going to be released. We talked about a variety of these different cytokines, and so they will then self-stimulate. They will cause proliferation, differentiation, but also we're going to start to help or recruit help from our other immune cells, so they can help out. Right? These helper T cells are going to activate our B lymphocytes. Similar scenario. We talked about this in the previous video. Right? Those B lymphocytes are going to proliferate, differentiate into plasma cells and memory B cells. And again, our plasma cells will produce those antibodies to help with whatever it is that is infecting us. And then our helper T cells are going to, again, activate another cell class, our CD8 cells, the cytotoxic T lymphocytes. Right? And then, of course, those cytotoxic T lymphocytes are going to go after whatever is the pathogen directly, right? And we'll talk about that in a moment here. And then finally, we saw that they will activate other cells in the innate immune system. And the example that I gave earlier is with our natural killer cells. So here's a wonderful picture, basically the effector response of our CD4 helper T cells here. These folks will release a barrage of various chemicals. The example here is interleukin-2. And then this interleukin-2 will self-stimulate these cells to proliferate, differentiate, and they'll help to activate other cells in our immune response, right? The cytotoxic T cells and our various innate cells. Okay, so speaking of the cytotoxic T cells, let's talk about their effector response. Same type of situation. We must have an exposure here to the antigen. Then we have now activated these cells. And what we'll see is going to be, again, in this scenario, we'll have activated T cells. And if it's a specific antigen that we've already been exposed to previously, our memory cells will then become activated and they will start to proliferate, differentiate, and these cells, instead of hanging out from a distance, they're going to go over to the infection site where the fighting is, where the battle is. And so they're going to get in there. They're our ground troops. So they get in there, they locate the infected cells. How do they do that? Those infected cells are going to have that displayed antigen on the cell surface receptor there, okay, our MHC class cells. And we will come in contact with those cells. And once that occurs, our cytotoxic T cells, the CD8 cells, are going to release those two famous granules, perforin and granzymes. And these are the substances that are going to literally punch holes into these infected cells and then, or these foreign cells, and then we'll pour in, all right, these cytotoxic chemicals into the cell and trigger apoptosis, that's cell death. All right, so this is a wonderful, wonderful system. It's worked for us for a long period of time. And so we'll see, all right, how this can turn the tide here during an infection, right? And so hence, right, we're dealing with specific cells. So, all right, these effector responses are going to work against these foreign antigens, right, that are associated with the various cells that are either the foreign cell or they've infected one of our cells and caused it to be classified as an unhealthy cell. Hence, that's why we call it cell-mediated immunity. So here's our picture here. We have our activated CD8 or our activated cytotoxic T lymphocyte comes in contact with the infected unhealthy foreign cell. And so it's going to produce that perforin, which is going to punch holes into the plasma membrane of the cell. And then we're going to pour in our granzymes there, which are pretty much going to cause the death of that cell. And we eventually wind up with apoptosis, which is absolutely what we want in this type of scenario. So last but not least, we have our B lymphocytes, the B cells here. Again, when they become activated, right, the majority of them are going to transition into plasma cells. And it's the plasma cells which are going to make and release the antibodies into circulation. So these cells don't necessarily have to be at the site of infection. They can be far away in a lymph node somewhere, and they'll just start doing what they do. They'll produce those plasma cells. Those plasma cells will then just release the antibodies into circulation here. And so the number of antibodies, it's really interesting. We have a 5-day lifespan for these cells, and we'll have millions of antibodies coming from one single cell. Right? And so again, it'll spill out those antibodies throughout your circulatory system for blood and lymph, looking for that specific antigen. So, you know, you hear a lot of times, especially with COVID, that people are going to get their antibody levels checked. And basically what they're doing is what we call an antibody titer. And what we're looking for is a specific antibody, in this case for COVID-19. And we're going to see and measure how much or what the concentration is of your antibodies, right, that is circulating through your blood. And so we have a certain range to determine whether you have a high amount, all right, or an appropriate amount, or where you don't have a real, you'll have like a low amount or something that's considered to be insignificant. Okay, so you've heard all this talk about antibodies. Let's discuss what they are. Antibodies are what we call immunoglobulins, and they're proteins, right? And they're proteins that are made, all right, against a specific or particular antigen. And so again, since they are antigen specific, their job is going to be basically to run through your body and tag those specific pathogens, all right, for targeting to be destroyed by your immune cells. We talked about various ways in which, all right, our antibodies do this. So these antibodies are wonderful, as we know, again, with current events, great for viruses, but not only that, right, bacteria, toxins that can be produced by bacteria and other microorganisms, and then, of course, yeast spores here. So these antigens fall under our humoral immunity. Right? Humoral immunity is basically anywhere in your body where you have fluid compartments. Blood, right, lymphatic system are going to be the predominant humoral compartments in your body. So what does an antibody or an immunoglobulin look like? Right? For the most part, right, they will appear as a Y-shaped structure. And I'll show it literally like the letter Y. Right? So in these proteins, right, we're going to have 4, not 2, but 4 polypeptide chains. Right? And these chains we identify as either heavy chains or light chains. And this has to do, again, when we're talking about the actual structure. Right? But what's important here is in our heavy chains, and I'll show you a picture here, right, we have a hinge region. And that's going to be located here around the Y. I don't even know why I'm doing that. I'll just show you the picture. All right. Here you can see, all right, our heavy chain is this big, thick yellow region. Okay? And then our light chain is this light kind of, I don't know what you would call that, kind of a yellowish area. But the hinge region is kind of where the bend of the Y is occurring, right there. Okay. So again, we have 2 heavy chains and 2 light chains. So what we'll see is, right, because of this structure here, we have 2 functional areas. Right? We have one, which is the variable area, and then a constant. Now that's important because the variable, and without getting into too much discussion about the variable areas, is basically our variable regions here are going to be part of the antibody in which we're kind of tweaking, okay, the structure to become better. It's like various models to cars that we've had over the years. All right, our current model is way better now for, let's say, I don't know, Ford Escape, all right, or a Chevy Malibu. Okay, so the model now, all right, I mean, I know it depends on who you ask, but the model now is going to be better present day than it was 10 years ago or 15 years ago if that model was still around. See what I'm saying? So these variable regions, right, we are going to improve, all right, the effectiveness of our antibodies. And this is important, right, because these variable regions are going to be the part that are going to come in contact with the antigen, and that's where our binding site is going to be. And so we're going to bind these antigens, important, you know, through these weak intermolecular forces. Right, so various way to actually bind these antigens can be, for example, through hydrogen bonds. All right, weak forces there, right, and that allows us, again, over time, and you'll see during an actual immune response period of time, the antibodies that you make initially during that period, right, aren't going to be as good as the antigens that you, or excuse me, the antibodies that you make at the end of that immunological response period. So let's just say, all right, I have COVID-19. I had it back in December of 2020, right? And so my antibodies that were made, all right, at the beginning of that response aren't going to be as good as the antibodies that I made at the end of that response, because it's these variable regions that your cells are tweaking, right, genetically to improve their ability to destroy whatever it is that that antigen is. All right, the constant region. All right, this is important because this is how we determine the biological function, right, for that immunoglobulin. And so we have, this kind of sets it into a specific class here. And so we have 5 major classes. We'll discuss these now, right? We have IGGs, IgMs, IGAs, IGDs, and IGEs. And that just stands for immunoglobulin G, right? Immunoglobulin M classes, right? And the letter just stands for the various classes here. All right, so here you can see basically, all right, our constant region, right here, is that bottom portion of the Y and part of where that Y bifurcates there. And then the variable region is usually going to be at the end, all right, of our light chains there. And again, that's the part we're going to be tweaking. That's the part we're going to be improving, all right, because we always want to get better. We want our immune response to get better, not worse. So let's talk about, before we get into the classes of our antibodies, all right, what do they do? All right, so there's a couple different actions for our antibodies. The first one is neutralization. Real simple. Literally, the antibodies are going to cover, remember all the antigenic determinants, right? The epitopes, where we have our fella right here, our antigen, and it has these little arms coming off. And those are going to be those antigenic determinants. So what will happen is these antibodies will swarm onto that structure and just cover that whole area up. And by doing so, it pretty much prevents it from creating infection in our healthy cells. The best way I can determine this, and if you want to look it up, you can. It's in the second Matrix movie, right, in which the main character, Neo, has to fight all these that they call Agent Smiths, and they're literally these viruses. And so there's a fight scene in which this one guy is in the middle of this playground, right? And then all these clones of the bad guy come running in, up to about 100 of them. And they just swarm on top of the good guy, thereby trying to neutralize him. And so that's what happens here, all right? So the antibodies just swarm on top. and every possible point of infection on the antigen gets covered up. So that prevents, all right, the virus or bacteria from being able to bind onto any cell receptor, making it useless. So that's neutralization. Right, then we have agglutination, what we call clumping. Right, these antibodies will create what we call cross links. All right, in which it allows, all right, a bunch of antibodies, all right, to attach onto these antigens, and it could be more than one antigen. And so what happens here is, if we're dealing with bacteria, okay, basically when you start to get this clumping, right, you're starting to make, all right, this grouping of structures more visible. So when you get, right, white blood cells or leukocytes, the T cells, right, just floating by, just kind of minding their own business, all right, they'll see this clumping and they can come over and investigate it, and they say, oh, okay, here we are, we have some antigens here, some foreign antigens, let's take them out. Our next action is going to be for antibodies is precipitation. All right, now with precipitation, all right, what we'll see, all right, again, similar, we'll have some antibody crosslinks that are occurring. And what will happen is, especially when we're dealing in the humoral compartments there, in lymphatic fluid or in our blood, right, when we get precipitation, when these antibodies start to form these cross links with the antigens, right, what we'll see is they will actually start to fall out of the solution. So they become insoluble. And so again, when they become insoluble, then they can be seen. Think of it, if you're soluble, you're kind of invisible, under the radar. Makes it harder, right, for when our white blood cells are going through on their surveillance, they cannot see, all right, these antigens. Well, the antibodies cause these cross-linking to occur, and then you get enough of them, they start to become insoluble and they precipitate out of the solution, and then that makes that whole complex more visible to the surveillance cells, and then they'll come in, and a lot of the time they'll undergo phagocytosis. A nice macrophage will come in there and gobble the whole thing up and say, problem solved. So of course, you might want to see it in a picture, always easier. All right, here's our neutralization. Okay, so here is, all right, our antigen, or this is a virus, for example. And so on the virus, all right, on the they call it the nuclear envelope of the virus, will have all these antigens here. And so all these antibodies come over and they cover up every single possible infectious point. So now this virus can't attach onto any of our healthy cells. And so we've pretty much rendered it, all right, useless. We've neutralized the threat. All right, agglutination. Okay, here we can see, right, again, we get what's called the cross-linking here, where our antibodies will start to cause this clumping phenomena occurring, commonly seen with the bacteria, right? And then we'll see this whole complex here will become visible to surveilling cells. And then we draw the attention of those surveillance cells and they come over and initiate, all right, the immune response to this area here. And then finally, we have our precipitation. Same thing, you have these small soluble antigens floating around, okay, in our lymphatic fluid or in our circulation, our blood circulation. And so the antibodies then will attach onto, all right, these particles, and then they will cause those particles then to become insoluble, thus coming out of the solution. And essentially, if you're into science fiction, all right, it's like a ship that has a cloaking device on it. It was once invisible to detection, right? No longer has that cloaking device working. And now, all right, these particles have become visible to our surveillance cells, and they come over and gobble them up. You know, for example, like I said, a macrophage comes in and just swallows things up. All right, so a couple more actions here for our antibodies. Right, so when we're talking about our antibodies now, all all right, for the further actions, we're going to be talking about what we call the FC region, which is that constant region. What is that? All right, that's this part down here. That's our constant region. That's what we're going to discuss here. All right, so don't forget about our complement system. It's a wonderful system. It's part, we discussed it when we were talking about our innate immune system. So here, our complement system comes into play through the process of complement fixation. So the constant region, which is the base portion of that Y on our antibodies, right? What we'll see is these guys, especially when we're talking about our IGGs and our IgMs, right? These guys are going to involve the complement. So those constant regions of both of those antibodies are going to bind onto the complement, and then that complement system will become activated, and then we can utilize parts of our innate immune system to help getting rid of those pathogens there. Opsonization here. Again, we talked about this before, right? We have our constant region, and what we'll see is, all right, through that process of opsonization, all right, we will then basically draw attention, all right, to the antibodies and whatever it is that they have bound onto, and we can get a target cell, right? One of our surveillance cells can come rolling by and then gobble up that whole, I guess you could say, not complement, but that whole antibody complex there that is attached onto the antigens. So we'll get our phagocytic cells, and those can be dendritic cells, they can, mainly macrophages. And so what they'll do is they'll come over, right, and they will gobble up, right, the offending agent there. And then, of course, finally, we have our natural killer cells, right, and our natural killer cells, right, will then, right, come in contact with the constant region here, and it'll cause the natural killer cells to release those cytotoxins, right, and those cytotoxins will then destroy whatever it is that that antigen is a part of. So I'll show you a picture here. Here we go. Here's the complement one. Saw this before. Okay, so we have our antibody comes in. It's going to, the constant region attaches onto the complement, causing that activation. And so then we'll bind onto our bacteria, will affect the complement system quite a bit. And so then that will start, we can start to get the MAC attack, all right, in which we start to punch holes, for example, into the cell membrane there of the bacteria. There's a whole series of things we talked about how complement destroys our pathogens in that scenario. Opsonization, same thing, all right? The antibodies will attach onto a phagocytic cell, all right? And it will activate that phagocytic cell then to attack the whole system or the whole complex will attach onto this bacteria. And then we can phagocytize the whole structure here. And then finally, we have our natural killer cell. Same scenario, the antibodies will attach onto the natural killer cell's plasma membrane through the constant region there, thereby activating the natural killer cell. The natural killer cell then will release, right, its cytotoxic chemicals, which are going to be porphyrins and granzymes, into the infected or unhealthy cell, and thereby resulting in apoptosis. So we've talked in earlier videos about how these processes work when we were dealing with the natural killer cells. So you can see antibodies play a large role here, right, in our cell-mediated immunity, but also it can help recruit parts of our innate immune system to help out, which we just saw. Complement, we can recruit our natural killer cells, and all right, even some of those phagocytic cells that we discussed earlier, you know, the big hitters, the macrophages there. All right, so earlier I mentioned that there were 5 classes of our immunoglobulins, or IgGs, IgMs, IGAs, IgDs, and IgEs. So let's talk about this right now. All right, our IgGs are the ones that make up the majority of the antibodies that are in circulation within our blood. All right, so we'll see, this is going to be the predominant type of antibody. So when we're talking about all these actions that we just mentioned, right, these antibodies, right, do all of that. These antibodies also, though, can cross the placenta, and that can be a problem, especially, all right, if mom's blood, right, is not conducive, or if there are antibodies in the baby's circulating blood, all right, that will have a specific type of reaction, right, to these antibodies here. So the baby's cells, the red blood cells that the baby has, might have antigens that these antibodies will attach onto. And it can cause what's known as hemolytic disease, in which basically, right, what will happen is these antibodies will bind onto the red blood cells of the fetus, and it can cause the cells to break down, and that could be a problem. IgM, right, is our next type of antibody. Now, this is the one, I remember I told you folks, right, the majority of antibodies have that Y configuration. Well, that's an awful picture, all right? But you saw the picture on one of the previous slides. Well, this one, all right, has this pentamer structure, which is you have like a centralized structure, and it has 5 different extensions coming off the center. And I think I have a picture of one. I don't. I thought I did. And I won't even try to draw it because it'll look awful. All right, but just imagine, all right, 5 Ys where the constant region all kind of anchor to one another. Right. So this one here is going to be great for agglutination because of that structure. Right, that pentamer structure there allows for more binding. And so when we're undergoing agglutination and complement reactions, this is a wonderful antibody to help with that. Right, and this is the antibody that we see, all right, when folks have, all right, a blood transfusion, for example, in which we have mismatched, unfortunately, blood typing, and so they'll start to see the rejection of that blood. This is the antibody that is going to be most responsible for that. IGAs are great because this is what I tell folks. These antibodies are going to be the antibodies that are going to have a lot of contact with things from the external environment. So you'll see that IgA antibodies are going to be produced by mucous membranes. And we know that the location of these mucous membranes are going to be around the exits and entrances to your body. Okay, so of course, we're going to see it all throughout, right, various regions that you produce mucus, but also in your mouth, because again, that's going to be coming in contact with things from the external environment. And then constantly our tears are being produced, not literally like tears where you're crying tears, all right, but that lacrimal fluid will have a high level of IGAs, all right, because you're constantly producing that, because again, okay, your eye is exposed to the external environment. And we also see it in large amounts in breast milk here. Again, the majority of your immune system is going to be found along, right, the GI tract, but not only that, your respiratory tract, because think about it, you are breathing air, which is coming from the external environment. All right, so the big role for these antibodies, right, is to help prevent pathogens from sticking onto and getting past our epithelial cells. Remember, one of the characteristics of epithelium is that it's tightly packed. So it's very difficult for things to squeeze in between the cells, right, because of some of those intercellular adhesion molecules, but also because they are tightly packed with one another. So then we coat that epithelium, all right, with something that contains high levels of IGAs, and so they help to prevent those pathogens from sticking onto the epithelial cells, but also those pathogens will try to burrow through the epithelium to get to deeper layers, while IGAs help with that. Right, so they do a wonderful job with the agglutination. Get that clumping action going, then we can take care of that business there. We get some of our phagocytes to get in there and take out those issues. IgD, right, these folks here are wonderful because they're going to come and help deal with or activate our B lymphocytes. Of course, you know what B lymphocytes do, right? So they'll come in and they'll help, all right, to activate those cells. So then we can get our B lymphocyte proliferation, differentiation to go on to help fight that, that pathogen there. And then our IGEs, right? We normally will see these in parasitic infections and allergic reactions. So these guys are going to stimulate the mast cells in your tissues, and then in our circulatory system, our basophils. And so they'll attract more eosinophils to the area. And we know that what eosinophils do, both eosinophils and our mast cells are going to release, right, the heparin and the histamine, and they'll help with that infection response there. Now, the nice thing about immunoglobulins is that our plasma cells, right, when needed to, can change from producing one specific type of antibody to a different type, and we call that class switching. And so with class switching, our helper T cells and the plasma cell, okay, are going to be involved. So the helper T cells are going to release certain chemicals that are then going to cause that plasma cell to switch over to a different class of immunoglobulin to help with whatever type of specific infection that we're dealing with. All right, you've heard me talk about memory cells, and memory cells, I'll be honest with you, haven't really been getting the just due that they deserve, especially when we've been talking about, you know, current events. You know, everyone's all concerned with antibody levels, you know, when we're talking about COVID-19. But what's important, just as important as antibody levels, is what about our memory cells, our memory T cells and our memory B cells? Because of course, when you come into contact with that specific antigen again, yeah, all right, maybe you were exposed to the Delta variant or the Omicron variant of COVID. Well, chances are really good, especially with the Omicron, right, that you will come in contact with it several more times because, all right, it has a high rate of transmissibility. Well, it's because of your memory cells that you are able to respond much quicker with subsequent reinfections. So we're going to kind of talk about this, right, when we're dealing with our memory cells here. All right, our memory cells are going to provide us, all right, with, all right, our immune response, all right, for future occasions, So when we're dealing with our immune cells here, memory results, and this is what I'm discussing, from formation of long-lived army of lymphocytes upon immune activation. Right. So here's what's going to happen. Right. We are going to initiate an immune response through adaptive immunity when we come in contact with that antigen. Now, the first time, all right, it's going to take us a little bit of time, all right, to respond because we've never seen this before. And so the time that it takes, all right, from first exposure, when you first come in contact with this antigen, right, until you start to produce, all right, an immune response, we call that the lag time. All right, that's the lag time. And so once you have that first exposure, then you will eventually form your memory cells. And those memory cells will be antigen specific for the next time, because most likely there will be a next time. Right? When I was growing up, chickenpox, I only got chickenpox once. Right? There's a good chance that I was exposed to it several more times, but because of my memory cells, it prevented me, all right, from being symptomatic and having any type of symptom or sign that I was infected. All right, so let's talk about this memory. So you've been infected, okay? For a very first time, you went through everything that we just talked about during an immune response, okay? We get that proliferation, all right? We get the differentiation of the cells, and then we have the effector response. So we just had that, all right? Months, weeks, months, maybe years go by. And now we come again in contact with the specific antigen. So we have those memory cells there. And so those memory cells, when they come in contact with that antigen, now we can get things done faster, way faster. All right, this is going to be our secondary response. Well, our secondary response, remember we have a primary and a secondary response, right? Pretty much the secondary response is going to be now, all right, to kill whatever it is that antigen is a part of. And so this response is way better than the first time you had a secondary response upon initial exposure. And so a lot of times you've destroyed whatever it is, the disease, the pathogen, way before you get any symptoms, all right, to present themselves. All right, the example here is measles. Okay, so you have, all right, your memory cells for the measles, you come in contact with it again, most likely you don't even know that you've been exposed because that virus, right, has been wiped out, right, by all the memory B cells and T cells. And we produced antibodies. We've brought in those cytotoxic, all right, T cells to come in. Maybe we've even had to recruit some of the complement, or not so much complement, but maybe we've recruited some of our phagocytic cells or the natural killer cells to come in. This is why vaccines are wonderful, right? Because they help us. This is important. They help us with developing memory. And we'll talk about that. We're going to talk about that. So here you can see our primary response, the very first time in which you are exposed to an antigen. Now this light blue area, that's our lag phase here. And you'll notice the primary response compared to the secondary response. Look at the difference, all right, in the lag phase. All right, the lag phase in the primary response, you know, is maybe 2-3 days. Whereas the primary response, or excuse me, the lag response for the secondary, the lag phase for the secondary response is much shorter. It could be hours. And you'll see the difference between, all right, the amount of antibodies produced from the primary response to the secondary response. You can see on the secondary response, your IGGs are off the charts. Compare that to the primary response, right? That IgG production is not so much. So you probably heard the saying, it's better to have it, not need it, than to need it, not have it. Okay, so when we're dealing with the secondary response here, it is boosted so much for your IgGs that you outpace the ability of the virus, for example, to proliferate and infect cells, because all these antibodies are rolling through, all right, your humoral compartments, just destroying any infected or unhealthy cell. And thus it cuts down on the ability for the virus to reproduce and cause its infection to be widespread. All right, so when we're looking at, if we want to kind of get a good idea, how do we measure the memory, all right, in our immune system, what we'll do is we can check your antibody titer, see how much of the antibodies are floating through your system there. And so we can compare that. All right, here you can see the antibody titers on the primary response are going to be much lower. See how small those are? And then you can see how much higher or bigger the antibody response is going to be during our secondary response. So those numbers will reflect, all right, how well your memory cells are actually working here. So here you can see, like I said, with the primary response, all right, that primary response can take days. All right, so anywhere from 3 to 6, floating usually around 5. So what we'll see during that time, during that lag phase, right, we have to see our antigens being detected by the immune cells. Then we have to activate the immune cells. Then they have to proliferate and differentiate. And then we have to start the effector phase. But what will happen during the lag is the first 4 parts there: we have to detect the antigen, then we have to activate the T cells, all right, and the B cells. Then those cells have to undergo proliferation and differentiation. So when our B cells then undergo that differentiation into plasma cells, then they'll start to produce, right? Initially, your IgMs will be the first antibody that get produced. Right, so if you see just IgM antibodies and not so much IgGs, then we know that we're still in the early phase here of the immune response. Okay, then our IgGs, those are the second antibodies that will appear. All right, so this takes some time, weeks, 1 to 2 weeks for this to occur. Now, when we're dealing with our secondary phase, right, when you've come in, or secondary phase, excuse me, our secondary response, right, now you've gotten over the initial response, months, years pass by, and then you've exposed to the same antigen again. Right, so now things happen on a much shorter, right, timeline here. So our lag phase is going to be much shorter thanks to the memory cells. They've come in contact with that specific antigen, and so we don't have to deal with, all right, all that stuff with detecting the antigen. We just did that, okay? And then we don't have to worry about activating and then trying to form memory B cells, or excuse me, memory cells and the activated cell line. We go right with the memory cells, the memory lymphocytes, okay, will immediately start to proliferate and differentiate. And so then those proliferated B cells will differentiate into our plasma cells, and we can start shooting out our antibodies. But in this case, right, we'll see our IgG antibody titer shoot right up, just like we see here in our secondary phase, or excuse me, secondary response. All right, so let's wrap up this chapter and discuss our active and passive immunity. Again, this brings in current events because you've been hearing a lot about active immunity. Okay, active immunity, there's 2 types. Okay, when we're dealing with active immunity, this is what happens when you come in direct contact with an antigen. So that could occur from natural infection. So you yourself were around somebody that had COVID and you got exposed to COVID, and now you undergo, all right, the immune response to COVID. Or you can get a vaccine. And then in the vaccine, all right, you'll have antigens in the vaccine, depending on which type of vaccine you get. All right, it's different for the mRNA vaccines, right? But you can have a vaccine, and then that will cause exposure to the antigen, and then you'll undergo the immune response for that. The important part about active immunity is you gain memory cells, all right, from active immunity to that specific antigen, whatever it is. You get the measles shot, you will now have, all right, active immunity, all right, against measles because you have formed memory cells that are specific to that virus. Now, passive immunity is when you get immunity from another individual. Breastfeeding is a perfect example, right? So mommy is breastfeeding baby, and in the breast milk, mommy gives, all right, antibodies, all right, to the baby. Right, so another example is the one here that we use, right? If you're bit by a snake, a lot of times they try to get you to the hospital where you hear they'll give you the snake venom serum. And so that's basically what will happen with passive immunity. We're going to give some sort of serum, all right, from one person to another. And we'll see that a lot too with folks that get, it doesn't have to be from a snake bite, but some folks they have developed, all right, they were asking people to donate blood specifically so they could get the antibodies from your blood plasma and then transfer those antibodies into people that got infected to help fight the infection. We saw that a lot here in the Greenville area. Folks were looking for blood donors to give the blood plasma if they've been infected with COVID, so they could use it to help others that were infected. Problem with passive immunity is we don't get memory cells. So it's good for the initial response, but it's not going to happen. You will not get, all right, that secondary response won't be as effective as it would be had you had active immunity. All right, so this figure here, 22, right, kind of breaks that down and shows you the difference between active and passive immunity. Notice though, all right, for our immunities, you have both naturally acquired, that's through, all right, the direct infection or natural. A lot of times in the news, you'll hear it being titled, you have natural immunity. And that's if you got it from somebody else or something else. Whereas artificially acquired is going to be through a vaccine. But they both fall under active immunity, which means that both people, whether you're vaccinated or not vaccinated for COVID-19, you will form your memory cells. Right now, there's big debate whether or not your antibody levels are going to be higher or lower, right? For folks that were vaxxed versus unvaxxed. That's not my point here. My point is that both forms, all right, will give you memory cells. So when you come in contact with that antigen again, okay, your secondary response will be much more effective, much faster. All right, so vaccinations, normally you are going to be given either the dead version or a much weakened version, they call it attenuated, all right, component, all right, of whatever it is that we're trying to vaccinate you against. But this form, a vaccination, this form of immunity is in the purpose trying to develop memory B cells. That's what we're trying to do. So again, if you're exposed to this, you'll have a much shorter response time. So vaccinations are really going to promote, right, the immune response in the humoral branch, which is pretty much your B cells. Right. The cell-mediated branch are the T cells. Nice thing about vaccinations, you know, for the most part, they can give you a lifelong immunity. Some do require a booster shot. All right, last part here of the chapter for our clinical view are the hypersensitivities. Now, this is important. So what is a hypersensitivity? Abnormal or exaggerated response of the immune system to an antigen. All right, so we have acute, subacute, delayed hypersensitivity. All right, acute happens very fast. Happens with me and my dog. I don't know what it is. I think it's something in her saliva, because anytime that I cuddle up with her, all right, any areas that she is able to lick on her own that touch my hand, on my arm or whatever, for whatever reason, I get some redness there. All right, so that's more of an acute hypersensitivity. Subacute takes a little bit longer, 1 to 3. So we're dealing with subacute hypersensitivities, right? This is going to be humoral immunity. Delayed hypersensitivity, again, 1 to 3 days, right? This will involve cell-mediated. All right, so hypersensitivity, another fancy way of talking about allergies. Right? And allergies are an exaggerated response, all right, to a non-infectious substance. Right? Peanut allergy. I know somebody that has a nickel allergy, the metal. I met somebody that had an aluminum or allergy. The darndest one was pineapple. Right? But anyways, in my life, I've met several people that just have various acute hypersensitivity reactions to various allergens. And so during these hypersensitivity reactions, right, we're going to see, all right, 3 phases here. We have sensitization, right, when you come in contact with whatever that allergen, and then we're now going to activate then the immune response, and then we will actually have the effector part of that response. And there's various symptoms that reflect, all right, these effector responses here, right, depending on the severity of the hypersensitivity reaction. Here in Greenville, South Carolina, we have tons of allergies, seasonal allergies, probably coming up here in about 3 or 4 weeks. We will start to see people running around with runny noses and watery eyes, snifflies all over the place, because we'll get all that pollen floating in the air. And some people have various reactions, okay? You can have issues with your breathing. We see that with asthmatics all the time. In fact, that's what, me as an asthmatic, I encounter that quite frequently, right? When allergy season is in high flow, it gets bad enough for me where I have to use an inhaler to counteract those symptoms that causes bronchoconstriction, right? Because of those allergens, they'll irritate the smooth muscle linings of my bronchioles, right? You've heard of the term hives, right? We just get itchy skin and you get these red blotches on your skin. If it gets bad enough, you can have vomiting and diarrhea, again, affecting, right, muscle tissue. And then you'll see in some cases, all right, widespread vasodilation and the inflammatory processes occurring. And if it's bad enough, that person can go into what we call anaphylactic shock. And usually they need to get a blast of epinephrine to help with that. Final clinical view is going to be HIV/AIDS. AIDS is known as the acquired immunodeficiency syndrome. Comes from a virus, right? The human immunodeficiency virus, or also known as HIV. This is a deadly virus, all right? And the fact that it kills our CD4 or helper T cells. And you saw everything that the helper T cell does, right? It's responsible for helping to activate, all right, your CD8 cells, your B cells, all right? It can also activate some of the cells in the innate immune system, right? It's widespread. So basically, this virus will infect these cells and destroy these cells, which is not good. So you can get this virus through sexual intercourse, drug use, needle sharing, okay? And we've seen it where an infected mother can pass it on to her baby through breastfeeding. So whether or not you start off by being HIV positive, you don't start off by having AIDS. AIDS comes later on in the progression of this condition, okay? So when you're first exposed to the virus, you are HIV positive. And so what we'll see over time, right, as this virus spreads and replicates and starts destroying your T helper cells, when we constantly take samples from you to determine what we call your CD4 or your helper T cell count. And if that count for those cells drops below 200 cells per cubic millimeter of our sample from you, then we give you the diagnosis of AIDS. And this virus really doesn't kill these patients. Right? These patients will die from what we call opportunistic infections or some sort of cancer. Right? It could be some, I'm trying to remember, there was a cancer that is very rare, except in AIDS patients. It's called Kaposi sarcoma. Again, it's a relatively rare cancer, but what will happen is what we'll see when the T helper cells count drops significantly, right? We're not able to stave off this type of cancer, and you see it in a higher incident rate. Various types of opportunistic infections, right? You can get some fungal infections that can cause death. These folks are fit into that classification of the immunocompromised. So they have to be very careful, all right, in trying to avoid exposure to certain pathogens here. All right, and a good way to help prevent, all right, contraction of HIV and AIDS is through safe sex, but obviously no needle sharing, okay, intravenous drug use, we try to avoid that. And obviously, if the mother is HIV positive, she's highly discouraged from breastfeeding her newborn. All those help to decrease, right, the spread of HIV and AIDS. Alrighty, folks, that's it for this video. I hope you enjoy. I hope you have a wonderful day, and I look forward to talking to you soon.